Okay, now we're going to go over the Biotechnology Study Guide. Number 1 says a blank is a genetically identical individual that is grown by taking a single cell from one parent. That would be a clone. Clones are going to be genetically identical, and they're going to be just like the donor, the organism that donated that genetic information. Number 2, which best describes the current use of genetic engineering? A says identifying hereditary diseases. The Human Genome Project is important for being able to identify hereditary diseases, but that's not officially genetic engineering because they're not manipulating the genes. They're just going in and looking at them. Vaccinating a child from measles, we're not really using genetic engineering for that just yet. Treating cancer with radiation therapy. They are working on some genetic engineering ways to treat cancer, but that would not be radiation therapy. Radiation therapy is radiation therapy. So C, making human insulin using bacteria. That is a current way that scientists and doctors are using genetic engineering right now. Number 3, a strand of DNA can be broken and a piece of another DNA molecule can be inserted, producing a modified DNA molecule. We looked at the gene splicing with the plasmid from bacteria the other day. So name 3 terms that refer to this process. That can be referred to as genetic engineering, recombinant DNA technology because you're putting in a new gene and recombining that, gene splicing, which means you're cutting that gene open and putting something in there. So genetic engineering, recombinant DNA technology, and gene splicing all refer to the same thing. Number 14, true or false, selective breeding, genetic engineering, and inducing mutations are all methods used to produce organisms with desired traits. That is true. Number 5, what process can be used to separate DNA fragments of different sizes? DNA fingerprinting is that term, or gel electrophoresis. They refer to the same thing, the cutting the DNA into different size pieces and then running them using electricity to run them through the gel and then looking at those banding patterns. That's DNA fingerprinting or gel electrophoresis. Number 6, true or false, recombinant DNA molecules are made by inserting a sequence from one organism into the DNA of another organism. And that is true. You take a sequence of bases that contains the gene, the desired gene, and you put it in there. And then that trait will show up in those new organisms. Number 7, lists the steps to cloning an organism. First, you remove the nucleus from an egg of an organism. And then the one that's going to be the surrogate organism. Then you're going to remove the nucleus from the body cell of the organism that's to be cloned. You're going to take that nucleus and implant it into the egg that you had removed the nucleus from previously. The cell will begin to divide and form an embryo. And the embryo will be implanted into the surrogate mother who will carry it and give birth to an offspring that is genetically identical to the donor animal. It's not going to be identical to the mother who's giving birth to it, but it's going to be identical to the donor animal. Number 8, how is the cell theory related to biotechnology? One aspect of the cell theory says that all cells come from other living cells by cell division. Okay? So all cells come from other living cells by cell division. This leads to the theory that you can modify a cell, a single cell, and then it will undergo cell division, producing generations of cells that have that desired trait. So all living cells come from other living cells by cell division. Number 9, what is gene therapy? Gene therapy is the use of genetic engineering to treat disorders. And sometimes disorders are caused by missing genes or maybe some bases are out of place that makes the gene not work properly. So gene therapy is using genetic engineering to treat disorders by inserting genes into the cells to replace missing or non-functional genes in the organism. Number 10, true or false, gene therapy. Viruses are typically used to deliver good genes in order to halt disorders. That is true. Viruses are very good at sticking genetic material into the genetic material of another cell. And that's just what we need them to do in the case of genetic engineering. So yes, biotechnologists often use viruses as a vector to deliver their genes that they're using to modify. Number 11, true or false, genetically altering cows to produce more milk is an example of why genetic modification is useful to human populations. Well, yes, having cows produce more milk would be useful to us. A lot of land is being lost to farms as people need more milk. Population increases. We need more milk. We need more meat. If you can produce a cow that can produce more milk, then you're going to have fewer cattle to give us the milk that we need. So they're taking up less space, eating less feed, fewer cows to milk. So that would be beneficial to human population. Number 12. There's 3 methods humans can use to get desired traits from organisms. Well, I started off with selective breeding before anybody knew anything about genes. So selective breeding is one way to get desired traits. Now, genetic engineering can be used. Inducing mutations, we have not talked a lot about that. That would just be introducing a mutagen into an organism that would cause a mutation that would give something a trait that you wanted to see in that organism. Inducing mutations is being used more and more to increase genetic variations in crops, like we talked about just a minute ago or earlier. That genetic diversity is very important to crops, because if everybody in a 100-mile radius is growing the same kind of corn, and a new fungus comes through that the corn is susceptible to, it's going to wipe out all the corn for a 100-mile radius. So all the crops will be gone. But if your neighbor's planting a different variety of corn, then the fungus might not jump over to his crop, and we would still have some corn there. So genetic variation is very important in survival of the species. So inducing mutations is being used a lot to increase genetic variations in organisms. Number 13, what charge is a DNA molecule? There's phosphate groups on the DNA molecules, sugar phosphate base. There's phosphates along that backbone. Have a negative charge. So DNA, because of all those phosphates, negatively charged, which means it will move toward the positive end of the gel electrophoresis plate. Number 14, there's 3 examples of genetically modified organisms. We have BT corn that we read about. It produces pesticides now because it's been inserted with a gene to cause it to produce pesticides. Golden rice has been modified to produce more provitamin A to help children, especially like in third world countries, prevent blindness, because they're getting that vitamin A that they would not normally get from their rice diet. And also we talked a good bit about bacteria that are modified to produce human insulin. And this helps to lower the price of insulin, increases the supply, so people with diabetes are not waiting on insulin and having to pay exorbitant amounts of money for it. Number 15, why is DNA fingerprinting useful? DNA fingerprinting can be used to identify someone. It can be used to identify the parent or offspring of someone, and it can also be used to help solve crimes. Number 16, as scientists have developed more productive crop varieties, farmers have switched from growing many traditional varieties to new high-yield varieties. For example, in Peru, the Indians once grew 500 native varieties of potatoes. That number has been reduced to about 20 major potato varieties worldwide. Why is it potentially dangerous to reduce the number of crop species of potatoes? And that's what we were just talking about with genetic diversity. The lack of genetic diversity increases the likelihood of a disease or disaster wiping out the entire potato crop in the area. If there were more genetic diversity, it would be more likely that at least one variety would be resistant and able to survive the disaster. Number 17, a gene for disease resistance in fir trees is transferred to pine trees using modern DNA technology. How might this be economically helpful to tree farmers? Now they can protect the pine trees and increase the crop yield for the tree farmers. They would be losing fewer trees to disease. Number 18, a DNA molecule that is produced by combining DNA from different sources or organisms is called recombinant DNA. We're recombining those DNAs. Recombinant DNA technology. Number 19, what does transgenic mean? Transgenic means that genetic material is taken from one species and inserted into the genetic material of another species. That would be like taking the gene, the human insulin gene from humans, and putting them into a bacterium, which is a whole other species. That would be transgenic. Or taking the cold weather gene from the fish and putting them into a tomato plant. We're going from a fish species to a tomato plant species. So that would be transgenic. So make sure you know all these and be able to read a DNA fingerprint. Being able to match those up, read a DNA fingerprint. That's it for the test next week.